of cell theory in the mid-1800s. And then last but not least, we're going to talk about the two types of cells that we know about. So here, let's get going. So start off, let's go back. Robert Hooke. Robert Hooke was the first one to use a light microscope. He was British. He was a British scientist, and he looked at cork pieces. So if you want to think, if your parents have like a wine bottle around the house, or if you've ever had like a beverage that had a thing that looked like kind of wood at the top, that's cork. So he would cut up a little of that and look at it under the very rudimentary microscope. And he noticed what he saw as small, tiny chambers, which he called cells, because they reminded him of the rooms in a monastery, which is a religious building where men or women would go and they study the Bible and be really religious, big in Europe. So going on, there was Anton van Leeuwenhoek. This is the guy, the father of modern microbiology, man of many firsts. So he was the first to use a microscope to study nature. His microscope was one of the first that was very powerful. The way you can think of his first microscope, so this thing right here, is take a pen or a pencil, and he was looking at what would be like right on the tip here. And think of it as like the peephole or to look outside on your door. He'd basically be holding that up to a little piece of glass like that, and he could see up to 200 times what would be visible to the naked eye. He was seeing. So he was the first one to see stuff. So his family was basically tradespeople, very normal people. His dad was a cloth merchant. And right around the age of 16, he started finding out that they would use these rudimentary microscopes or viewing devices to see the thread count on a piece of yarn or fabric that was going to be used. And if you know anything about good sheets and stuff, you know that the higher the thread count, typically, the more soft and more sought after that type of cloth is. So if you could see how good a cloth was at really close up, it would give you a leg up on the other person if you're buying or selling this stuff. So he got one right around what is suspected to be about 16 years of age, and he started making observations. So he first saw these different organisms, things around you would have around your house or in your world, and he was seeing them at like 200 times, and he started making his own microscopes. And he was a really good illustrator, so he was drawing what he saw, and he was the first one to look at different things. And he would send these pictures and these drawings off to the Royal Society, which was a very well organization in London. And they were like, wow, this is pretty cool stuff. But they didn't believe him. They did not believe him. So they dispatched a couple people across the English Channel to Europe to go find Anton and see what he was doing. So he was one of the first people to look at pond water. And these are a couple of his other firsts. He was the first one to look at muscle fibers. So, let me see your guns. Let me see these. He was the first one to look at that under a microscope. Bacteria, sperm, red blood cells that run throughout our body, crystals, things that look real pretty. And he was the first one to see blood flow in capillaries, so the very small part of your arms and all over your body where the blood goes to and from the heart. He was the first one to see all that stuff in action using one of these very rudimentary microscopes. So first microscopes basically looked like upside-down telescopes. So if you ever look at a telescope and flip it upside down, instead of seeing stuff that's really far away, you're seeing it really close. So here, basically, instead of looking this way out to the heavens and looking at like Jupiter, you're looking from this way down at the really small stuff, and it's very amplified. Now, toward the mid-1800s, you had a microscope that looked, I'm going to say, pretty close to the ones you're going to use in class. So it's got the barrel. You put whatever you're viewing here on the stage. And basically, instead of having a light to magnify or, give definition of the image you're looking at, they would bounce light off of a mirror and then up. So basically, outside of a modern-day light source, not a lot has changed. So important discoveries along the way. Matthias Schleiden came up with the discovery that all plants are made of cells. So he was the first one to really get that they're kind of square and boxish, and they are small compartments, and they're all made of cells. Theodor Schwann, all animals are made of cells. So we're kind of narrowing it down. And then Rudolf Virchow, all cells come from pre-existing cells. So we talked about this in the 8 characteristics of life. In order to get life, it needs to come from something that's living, okay? So that led up to cell theory. All living things are made of cells. If you have one, it's unicellular. 2 or more, it is multicellular. Number 2, cells are the basic unit and structure of function in all living things. So we learned about that from the very get-go of the school year. It has to have a cell to be considered living. If you get more, it's still living. New cells are produced from existing cells. So to get the chicken, you need to have a chicken first, okay? So you cannot start anything without having a cell to start off with. And now we're going to get to the parts of a microscope. So first thing, pretty self-explanatory, is going to be the eyepiece. This is the top. This is where you're going to look through to see the image that's being magnified. This is going to magnify it 10 times or 10x. Next is going to be the coarse adjustment wheel. This is for making very big changes. Typically, this will be the big wheel up there. Then there's going to be the fine adjustment wheel. This is for making very small changes. So typically you will put a slide under here and narrow it and get it into view using the coarse adjustment wheel. Then you will start using the fine adjustment wheel to make small changes. These are your objective lenses. These magnify the image. So typically you will have 3 of them. There's 4x, 10x, and 40x. So to get magnification, you take 10 times whatever the objective lens. So like the lowest one will be 40x. So 10 times 4 is 40. Next would be 10 times 10 for 100 times bigger than what you're looking at. And then the microscopes we're going to use at school, most biggest amplification or magnification you can get is 400x, which would be 10 times 40. Next will be the neck arm. You will use that to help carry the microscope to and from where you're going. The stage, so if you want to think of like singing and dancing, you would do it up on a stage. That's what you're going to put your slides or what you're trying to look under on the stage. It'll have clips or something to help hold the slide. There's going to be the light, which you want to make sure is on so you can see what you're viewing. Then you'll have the base. That's what it sits on. So when you're carrying it, you'll hold one hand on the base, one underneath, carry it around. And then the diaphragm. This adjusts the level of light. So if you can't see it, adjust it. It'll let more or less light into the viewing area. You will have a quiz on this in a day or two before we start doing lab. Finally, the 2 types of cells that we're going to be talking about. There's going to be eukaryotic and prokaryotic. So prokaryotes and eukaryotes. Biologists have divided cells into 2 different categories. Prokaryotes have no nucleus. These guys are very, very simple organisms. Then there are eukaryotes. These are more complex. So, prokaryote cells. These have no nucleus. So the way I like to think of it is they are kind of, everything's mixed in here. So they got the DNA and the organelles all together mixed in. So think of it as like you had leftovers and you just threw it into a bag and then threw it into the refrigerator to keep cool. So spaghetti, beans, whatever, you just threw it in a bag, threw it there. You didn't try to box it up, put it away. So they will have a cell membrane, which is the outside of the cell. Keeps the stuff from inside the cell from getting out and stuff from outside getting in. They'll have the cytoplasm, kind of like the middle of the body. It'll have ribosomes. It'll also have DNA. The DNA is just kind of mixed in with everything. Last but not least is eukaryotic cells. These have a nucleus. So these are more complex. So plants and animals are eukaryotic cells. Example, plants, animals, fungi, and microorganisms. So we will basically be studying more eukaryotic organisms this year. These guys can come in many different shapes and sizes, okay? And that's what we're going to look at the next couple of days. Got any questions? Let us know.